All right, let's all please welcome Ralf Goericke from Scripps Institute of Oceanography, and he'll be talking about climate forcing of phytoplankton in the Southern California current.
Thanks for inviting me. I'll be sharing some recent data that we have from the CalCOFI time series. Just, as an alert, I will not be really talking about the California Current Ecosystem Long-Term Ecological Research Program. Mark Ullman did that here a few years ago. So with that, I'll first be introducing you to the CalCOFI program, and then I will look at some interesting recent data. So our study area shown here is located in it's part of the California, of the Pacific, North Pacific Gyre. The California current flows down the coast, meandering, spawning eddies, and this is also a region of very high mesoscale activity, which makes it a really interesting area from a physical point of view, but a really messy area biologically, because it is really a confluence of water masses from many different places. This is the conceptual diagram which is guiding our thinking about the system. The system is dominated by winds that along the coast induce coastal upwelling, in the offshore current-driven upwelling, shown here, in the nutrients in the form of blue arrows get pumped into the euphotic zone. At the same time, iron is delivered to the system from the sediments and also from aerosols. The nitrocline depth in our system, nitrocline depth, the depth where concentrations of nitrate reach values of 1 microgram per liter, is a really important driver of productive standing stock of biomass. And we envision our food webs as dominated by larger organisms in the coastal upwelling systems and smaller phytoplanktons in the offshore systems. So the CalCOFI program, and that stands for California Cooperative Oceanic Fisheries Investigations, and don't blame me for that acronym, it was founded in 1947 in response to the collapse of the sardines off California, which were heavily fished during the 1940s. And the question, as always in those cases, that arose was this collapse driven by overfishing, or was it a natural cause? Instead of solving a problem, a committee was put in place, CalCOFI, to study the problem. And that's what we have been doing ever since, with great success in many areas. And the sardine problem was also solved. But let me now go today. Today, CalCOFI is a partnership between the California Department of Fish and Game and NOAA Fisheries and the Scripps Institution of Oceanography. The costs are borne primarily by NOAA, Scripps, and Cal Fish and Wildlife is contributing a little bit. The key components of CalCOFI are the Ocean Observing Program, an annual conference, a journal, CalCOFI Reports, which may be shut down soon, and experimental field work. So, these days, and we have been doing this since 1984, we occupy a pattern of 75 stations of Southern California, up to 500 kilometers offshore, during the summer and fall. During the winter and spring, we occupy this pattern to better characterize distributions of sardine, anchovy, larvae, eggs, and adult fish. So these are the parameters that are measured regularly on the CalCOFI cruises. Covering physical chemical parameters, the standard ones, lower food web planktonic measurements, and measurements on small pelagics. It's really important to note that a variety of ancillary programs have sprung up around CalCOFI, greatly enriching the data set, but working within the CalCOFI framework. The CCLTR program, which is augmenting CalCOFI measurements through measurements of the geochemistry, biogeochemistry, and of planktonic communities. We're monitoring the inorganic carbon system, though we are looking for funding to continue this right now. We are taking samples for genomics in collaboration with the Craig Venter Institute, and we have had seabird observations and marine mammal observations for a few decades. So these are the timelines of the time series that we have. Some of these date back to 1949. The standardized CalCOFI pattern was adopted in 1950, and since 1984, when the CalCOFI field program was reorganized, we have been occupying these 66 CalCOFI stations on a regular basis and have made consistently measurements of primary production, nutrients, chlorophyll, etc. So I will primarily focus on the 1984 to the present time series. And again, really important, in 2004, with funding from NSF, the CCLTR was started. So this is the 66 standard CalCOFI stations are shown in blue here. And CCLTR samples on all those stations too, but at the cardinal station shown indicated by circles here, sampling is more intense. We have coastal stations. We have spray gliders that operate along lines 80 and 90, 2 moorings, one located in the California Current proper, the other one located in the coastal upwelling region. Craig Smith has a benthic station slightly north of our sampling domain, and remote sensing is augmenting our observations. So we have a complex but complementary system of observations which allow us to characterize what's happening off Southern California in incredible detail, spatial and temporal. So all these are programs ancillary to CalCOFI. They are not CalCOFI. So the data products that we routinely produce are, of course, temperature, nitrate, chlorophyll shown here to illustrate the large gradients in these properties as you go from the coastal areas to the offshore. And also the ranges of value. For example, chlorophyll A ranges by almost 2 orders of magnitude from the upwelling areas to the offshore areas. Sorry. Another data product are fish eggs, which are collected on the cruises using an automated system, which allow us to monitor populations of spawning fish, sardines, anchovy, and jack mackerel in this case. And observations such as those are the motivation for using a large spatial footprint for the CalCOFI programs. Just an example of how great it is to have really long time series. Shown here are concentrations of oxygen in our area in green from 1950 to 2010. This was published some time ago, showing that the recently low concentrations of oxygen are not unprecedented, but they were also observed during the 1950s. But more importantly here, shown in blue is a proxy for the abundance of mesopelagic fish, showing dramatic variations in their abundance with time, co-varying tightly with oxygen. So changes in concentrations of oxygen at that depth interval imply changes in the hypoxic boundary. And the changes of this hypoxic boundary reduce the habitat for mesopelagic fish fauna during the day, which renders these more vulnerable to visual predators. So Oxygen changes affect habitat, contracting habitat affects abundance. So this is an example for why long time series are really great to have. So now let's look at some data here. So the objective that we all or one of the objectives that we all have who do time series work is to predict the response of the ecosystem to global climate change. We are still far away from that, of course. So the questions that I'd like to address is, how does the California Current ecosystem, CCE, respond to changing North Pacific climate? Does the response of the CCE to climate forcing change with time? So we have seen the climate indices, the PDO, and I've determined change points here, statistical properties of the time series which indicate where the strongest changes of the mean are. I'm not talking about regimes, this is just a statistical property. And this is the NPGO. The NPGO is interesting because when it was first proposed, it was stated that it explains variations of sea surface salinity, nitrate at depth, and chlorophyll A in the CalCOFI region. Explains. However, recent observations of sea surface salinity, and these are now data, like most of the data I will show, which are based on averages across all 66 standard CalCOFI stations for each cruise. So the recent sea surface salinity, red, does not seem to co-vary that nicely any longer with the NPGO. And indeed, the two are still significantly correlated, but weakly so. But more worrisome, the residuals have significant autocorrelation, which tells you that you can't carry out such a regression. What one has to do is take the autocorrelation in the salinity time series into account as well and model it using such an equation. When you do that, when you fit an ARX10 model, and time series people will know about these, to the data, you find that the autocorrelation is indeed extremely significant. But the correlation between salinity and the NPGO is no longer significant at all. So, to conclude this, CalCOFI domain salinity over the last few decades is no longer explained by the NPGO. The NPGO likely reflects the integration of multiple physical forcing functions, some of which affect CalCOFI sea surface salinity, others not. Identifying these forcing functions is important. So this results adds to the growing body of evidence that cautions us to use ocean climate indices such as the PDO and by inference the NPGO as predictors of ocean state. And the practice that Jennifer showed, using multiple indices, that is what is required from us. So let's look at some other aspects. How does the CCE respond to basin-wide forcing? Does the response change with time? And response is the key here. And so I'll be focusing on relationships between physical forcing, stratification, nutrients, and phytoplankton biomass. So shown here is the temperature time series, which just like off Newport reflects the strong ties to basin-wide forcing. Again, the change points of the time series coincide roughly with the change points in the PDO time series. Shown here are nitrocline depth, mixed layer nitrate, mixed layer chlorophyll A. And these three variables likely represent a causal chain. Nitrocline depth affects mixed layer nitrate, mixed layer nitrate, mixed layer chlorophyll A. So these are cruise averages. So you average out temporal variability in the system that you have temporal variability on the order of days or so. So are the responses to forcing stable during the three different time periods? So by by simply plotting the cube root of nitrate, I choose that transformation to get variables log normal, normally distributed, against nitrocline depth, we see for 3 different time periods that coincide with the time periods of the change points, pre-1999, 99 to 2014, 2014 to the present. We see that the pre-1999 time period, blue, has a distribution that differs a lot from the 1999 to the 2014 distribution. So this suggests that the response, or that the mechanisms that mediated the response of nitrate to mixed layer nitrate, to nitrocline depth, changed between these 2 time periods. And interestingly, the data from the recent time period are more similar to the pre-1999 time periods. So the same analysis for log chlorophyll A and nitrate does not show such large differences between these time periods, but there are likely other factors other than nitrate affecting chlorophyll A concentrations. So nitrocline depth, mixed layer nitrate, and mixed layer chlorophyll A are strongly responding to basin-wide forcing, ENSO and PDO-ike. The pre and post 1999 responses differ, suggesting that the mechanisms coupling these differed. So let's look at response data such as those that I've shown here, and that's one of the main reasons that I did so. Those should be used to gauge the quality of model outputs rather than simple comparisons of property maps, real data maps, model maps. So that's the main point that I'd like to make here, that such observations from our time series should be used to inform models. So let's quickly look at some spatial patterns. I'm using EOF analysis, and you separate this time series has a spatial and a temporal component, and you can separate these into principal components to get the temporal component and EOFs to get their spatial footprint. Let's quickly do this. I'm running out of time. This is the spatial footprint for chlorophyll A here, and the time series shows a really strong response of the chlorophyll time series to the recent warm anomaly and the El Nino. Interestingly, the change points of this time series no longer correspond to the change points in the PDO time series. And to me, that suggests that this here reflects forcing, which may be contained in the PDO time series as part of the multiple forces, but it no longer adds up to produce the PDO-like pattern. So suggesting too that the PDO does not reflect forcing in the system well. The spatial pattern for nitrate has a strong footprint in the upwelling areas, and the time series, the PC, is similar to the chlorophyll time series. Nitrocline depth has a strong footprint in the offshore areas where the California current changes stratification and upwelling, open ocean upwelling changes its depth. More importantly, let's look at how these spatial footprints change between time periods to gauge if the response of the system changed. So the spatial patterns for chlorophyll A do not differ dramatically between these 3 time periods. The same is somewhat true for nitrate, except that during the pre-1999 time period, the spatial pattern was concentrated off more than it is during the recent time periods. And the same can be said for nitrocline depth, the spatial patterns are all very similar. So, the spatial patterns did not differ greatly between time periods. So this suggested the spatial patterns of the forcing that affects controls the first EOFs of these properties did not change dramatically between these time periods either. So to summarize, the CalCOFI program has evolved from a fisheries monitoring program in its early stages into a comprehensive ocean observing system. Its long-term time series allow us to address climate questions on decadal scales. Relationships between basin-wide climate indices and local properties can be highly variable, in particular if the basin scale indices integrate multiple and diverse processes. State variables respond strongly to basin-wide forcing. The response of forcing changed over time for the case of some variables, not others. So we need to take into account, we need to consider that the mechanisms which mediate responses change between these time periods, and how that occurs we need to find out. Thank you. Yeah. Yeah.
Hi, I'm trying to understand a little bit better in biological terms what the changing response means and what you were seeing with the nitrocline depth. So what you're seeing is that now you get less chlorophyll for a given size of nitrate input?
Yes.
Okay, so can you speculate on what that could be caused by?
It is the response of phytoplankton biomass to the rate of nutrient delivery to the system. On the one hand, does that answer it? And it also affects other parameters of our system which I didn't show, such as phytoplankton size structure and so forth.
Okay, thanks.
Hi, Ralph.
Yeah.
Patty Matrai Bigelow over here.
Yeah.
So it's fabulous to always see how it's progressing.
Pardon me?
I say that it's fabulous to see
Yeah.
how the
Oh,
data set is progressing. However, now you have nitrate. This is only for nitrate. It started a long time ago, a lot of those data, and by now you probably have also developed some relationships to the parameters that have been physical parameters in nutrients that were measured since 1950. If you were to plot those relationships, for example, chlorophyll to temperature, and you were to expand to the entire time series, what would it look like? In other words, taking advantage of those data, since there have been other analyses up to 89 of the CalCOFI data set done in a similar way to what you presented today.
For some parameters, we can do that. So we can back calculate the pH using concentrations of oxygen, temperature, et cetera, But, to back calculate parameters such as phytoplankton biomass, for which we don't have consistent measurements prior to 84, I'd be cautious, because one of the results that I showed is that we cannot assume that the relationships upon which we base these predictions are stable. And that's why I went through this exercise to find that out.
Ralph, over here. Thanks for that overview. Just before the break, we heard about the broad-scale surveys along the Oregon coast, going up north. Granted, they have not been carried out that long, but CalCOFI used to extend all the way to the probably Canadian border and further down along Baja as well.
Yeah, it went.
Yeah. So my question is, some of these correlations may also be informed by linking with these other observing initiatives.
Yes. Just that would be great. One of the problems is that lots of those are carried out by NOAA, and they like to sit on their data.
Ralph, nice talk. Barney over here.
Barney.
Barney Balch-Bigelow. How about silicate and silicate nitrate balances? Presumably those have been measured since time immemorial in CalCOFI. How have they changed?
They are varying quite a bit. For example, in the around 2005, we had a low, for a few years, really low concentrations of silicate, and the silicate nitrate ratio recently has changed in an almost stepwise fashion. What is driving those, is not that easy. We published a paper, Steve Bograd, with the lead author on that in 2015. Our region is thus impacted by water from about 4 different directions: California undercurrent, the North Pacific gyre, the California current. The California current changes its composition. That's a really hairy problem to figure out what exactly is going on. But it's really interesting.